morning, everybody. My name's Abby Hodson, and this summer I worked on creating a coupled hydrodynamic biogeochemical numerical model of Shelburne Pond, and I did this under the guidance of my research advisor, Dr. Clelia Marti. So just to give everyone a quick overview of what I'm going to be talking about today, I'd like to start with the inspiration for this project and the problems I hope to address with it. Then we'll dip into sort of the methodology and the timeline that I followed, and then we'll look at some of the actual data that was produced by this model. When I was imagining what I wanted to do this summer, I started off by thinking about a problem I was passionate about, which is eutrophication. And for anyone who isn't familiar with the term, eutrophication is an environmental process that occurs when stormwater loaded with nutrients like nitrates and phosphates stimulates plant growth in aquatic ecosystems. And specifically, algal growth is what we're worried about here. And even though there can be perfectly healthy concentrations of algae in freshwater ponds and lakes, what happens is that the excess nutrients in the water cause the algae to dominate the ecosystem and form a surface scum which prevents sunlight from penetrating through to oxygenators like plants at the bottom of the pond. And as the algae start to die off, they require oxygen to decompose. And because fewer and fewer organisms like fish can survive in low oxygen conditions, these organisms also die off and also require oxygen to decompose. So you can see that when nutrient-loaded runoff enters our waterways, it actually kickstarts this positive feedback loop of lowering the oxygen content in water and lowering the biodiversity in that water. And this cycle continues until your pond or lake is essentially reduced to a hypoxic puddle unsuitable for life. So eutrophication is a highly complex economic, social, and environmental issue, and a lot of people are working towards a solution right now. But it's so complicated that there's not going to be a one-size-fits-all solution. So I thought the best way that I could contribute to finding a solution would be to create something that people can use to better understand and visualize this process, to know what the physical, chemical, and biological drivers are, and actually understand what's happening when we talk about eutrophication. And the best way to do this is with what's called a three-dimensional aquatic ecosystem model, or an AEM 3D model. This specific type of model is produced by a company called Hydronumerics in Australia. And it's basically a library of algorithms that simulate hydrodynamic and biogeochemical processes. And a user can upload relevant files and tweak that code to their specific project. So what I did this summer is I created one of these models for Shelburne Pond, specifically with the hope of modeling algal blooms. And as you can see from this little timeline, the first half of this project occurred this summer, and that was implementing the model and then validating it using actual measurements. And when I say validate, I mean comparing the predictions of the model to real measurements to see just how far off we are. The second half of this project is ideally going to occur this fall as part of my honors thesis, in which I hope to actually implant an algal sensor system in the pond and have it feed data directly into the model. That way we can predict blooms in real time and automate the model validation process. And all of this is happening with the ultimate goal that this model can be an asset to the town of Shelburne, something that can encourage better informed environmental legislation and education as well. So like I mentioned before, I chose Shelburne Pond specifically for this project because eutrophication is highly prevalent in Vermont. We hear about harmful algal blooms in Missisquoi Bay, Burlington Bay, Shelburne Bay, and Shelburne Pond is a site that has historically struggled with managing cyanobacteria blooms for decades. So I wanted to create something for this smaller scale body of water that could potentially alleviate this issue. A little timeline of how exactly I implemented and validated it. I started with a thorough research review on modeling and algal growth to really understand what was happening. Then I found and formatted the bathymetry of Shelburne Pond through mashing together several different data sets from student projects back in 2010. Then I looked at local weather monitoring stations and projects for the data that I needed to force the conditions for the biogeochemical process under the water to occur. Then I edited the core code of the AAM 3D model to only simulate the processes that were relevant and that I cared about and output the relevant variables. Then I validated it using in situ measurements from a buoy monitoring project that was set up by Dr. Stockwell in the Rubenstein School. So like I mentioned previously, validation occurs in two steps, the first of which being hydrodynamic validation, which is basically only running the physical portion of the model and looking at specific variables to see how far the model predictions are off from actual measurements. And one of the variables that I looked at to prove this was temperature throughout the water column. So if you look at these graphs on your left, you can see that the temperature simulated by the model closely follows the temperature actually measured by a thermistor chain in this pond. And this means that we can essentially trust the physical portion of this model. And if we perform statistical analyses between these two data sets, we see that the differences are statistically insignificant. And we can move on to the biogeochemical validation, which is a little bit more tricky than the hydrodynamic validation. And the reason that it's difficult is because the data set that I'm using to ensure the accuracy of this model, the data from that buoy in Shelburne Pond that I was talking about, measured something called phycocyanin fluorescence rather than the direct concentration of algae in the pond. And phycocyanin, for those who aren't familiar, is a pigment that's found in a specific type of algae, cyanobacteria, which fluoresces under specific wavelengths. And though it's correlated with the actual concentration of cyanobacteria, we can't directly convert between these two variables confidently. So the best we can do is look for peaks in phycocyanin fluorescence and peaks in cyanobacteria concentration. And if we look at these charts on our left, we see that cyanobacteria concentration and phycocyanin fluorescence peak at roughly the same time, around the 230th day of 2015, which would essentially mean that we're getting a cyanobacteria bloom midsummer, which checks out. But we need to look at more biogeochemical variables that we can actually perform statistical analyses on before we can fully trust the results of this model. So with that in mind, we can start looking at some of the simulations. So the movie on your left is a simulation of cyanobacteria on the surface of Shelburne Pond, and the movie on your right is a profile view of that same pond. So as we look at this blue, we see that it starts off pretty slowly, pretty low concentration as the water is cold. And then as snow starts melting and storms start happening and mixing occurs, we start to see concentrations rise. And once we get into the summer months, July, June, August, we see some actual blooms happening, which is the orange and red on your left. And as fall starts to occur, it's going to wind back down again. But basically, cyanobacteria thrive in warm, stagnant waters. So it tends to occur in July, August, and it's also accelerated by climate change. And if you'll look at these charts, you'll notice that this model is running for the algal bloom period in 2015. And the reason that I chose 2015 is because this is the year with the greatest data availability. However, the second half of this project, which I plan to pursue in the fall, will incorporate data from recent years and also live data from the pond, so we can actually look at these blooms in real time. So I need to give a big thank you to my advisor, Dr. Clelia Marti, who advised me throughout this process. She's been in Australia this whole time, but has miraculously been meeting with me and emailing me regularly, so I really appreciate that. Dr. Jason Stockwell, who set up the buoy monitoring project in Shelburne Pond and provided me with the data I needed to ensure the accuracy of this model, and all of the Barrett interns, faculty, and the foundation for supporting this project. Here are my references. Thank you all for listening.